OK, bonjour tout le monde. On va débuter. Donc, merci tout le monde d'être là pour cette conférence de l'IBIS qui a lieu sur Zoom. Et aujourd'hui, on a comme conférencier le professeur Edel Pérez Lopez, qui est professeur adjoint au département des sciences des plantes ici à l'Université Laval. On avait invité Dr. Lopez à donner une conférence au mois d'avril l'an passé, mais à cause de la pandémie, on a dû repousser sa présentation. Et donc, maintenant, il est aussi membre associé de l'IBIS. Puis, je pensais que cet automne, c'était une bonne occasion de l'inviter pour que tout le monde à l'IBIS puisse le connaître. Donc, comme je mentionnais, professeur Pérez Lopez est le professeur adjoint au département des sciences des plantes. Il vient tout juste de débuter. Il a fait ses premières demandes de subventions pendant l'été et cet automne. Et il est membre aussi du Centre de recherche et d'innovation sur les végétaux. Et comme je mentionnais un peu plus tôt, membre associé de l'IBIS. Dr. Pérez Lopez a fait sa formation dans 3 pays différents. Donc, il a fait un stage postdoctoral, en fait, dans 4 pays. Un stage postdoctoral ici au Canada, en Saskatchewan, et un autre stage postdoctoral auparavant en pathologie des plantes à Auburn University aux États-Unis. Et sa formation doctorale a été faite à l'Université du Veracruz au Mexique. Donc, c'est là qu'il a obtenu son doctorat. Et originalement, il vient de Cuba. Donc, il a fait sa formation de premier cycle à l'Université de La Havane à Cuba. Dr. Pérez Lopez s'adapte ultra rapidement au climat et à la société scientifique québécoise. Il est déjà directeur de la Société de protection des plantes du Québec. Il est aussi coprésident du comité d'éducation de la Société canadienne de microbiologie. Donc, déjà, en commençant sa carrière, il occupe des postes de leadership. Donc, je pense qu'on est chanceux de l'avoir à l'Université Laval et de l'avoir comme professeur associé à l'IBIS. Et puis, je vous laisse la parole pour votre présentation.
Merci beaucoup et merci pour l'invitation. Je suis très heureux et je vais parler en anglais parce que j'apprends français, mais je pense que l'année prochaine, je vais parler en français. If you invite me again. So the title of my presentation is the clubroot pathogen, a master manipulator of plant immunity. So I wanted to talk a little bit about, even when Christian already introduced me, but yeah, I did my bachelor in biochemistry in Havana University, and also in the National Institute of Biotechnology. So I work in Cuba. We have to develop a undergrad thesis, and my thesis was with dengue fever virus, completely different to plants, but it's basically because plant research is not that well funded in Cuba. So I wanted to learn molecular biology and basic techniques. So I decided to go here. Once I finished my undergrad, I went to work as a researcher first in the National Institute for Plant Health. And then shortly after, I went to the Institute for Tropical Fruit Crops in Havana. Later, I did my PhD in the University of Veracruz. And during that time, I was able to visit Canada for the first time to do part of my PhD. I came to Agriculture and Agri-Food Canada in Saskatoon. Later, I visited again Saskatoon and I went to US as a postdoc for 1 year to later come to University of Saskatchewan again for a postdoc, and now I'm here as an assistant professor at Université Laval. In Cuba, it was really important for me, the research I did, because I had my first encounter with phytoplasmas. Phytoplasmas are unculturable plant pathogenic bacteria, members of the genus Candidatus phytoplasma. It's a really complicated pathogen to work because it cannot be cultured. Something also important about phytoplasmas is that they are responsible for big economic losses. And this is still a debate. Many people think that not really, but for example, here in Canada, we had a big outbreak around 2010 in canola crops in Saskatchewan. So that didn't happen again, fortunately, but it's related a lot to the presence of the insect. Phytoplasmas are transmitted by leaf hoppers mainly. So if we have a lot of cicadel, it's in French, we will have a lot of phytoplasma and then the losses can be huge. Well, in Cuba, I was able to publish my first two international papers in New Disease Report. It's a small journal of the British Society of Plant Pathology. And I found phytoplasma in macadamia trees. This is not a common fruit in Cuba, but we had a collection of exotic fruits, and we found that in these macadamia trees, we had phytoplasma. And then the next paper was about sapodilla trees infected with phytoplasma. Sapodilla, probably many of you don't know this fruit, is really tropical and it's also, we don't see it too much in Cuba for tourism, but it was important, it's important for the economy. So after my time in Cuba working with phytoplasmas, then I decided to go to Mexico, and I was really lucky that I had the opportunity to keep working with phytoplasmas. Now I was working with a corn. There is a really devastating disease affecting corn in Latin America, and it's caused by phytoplasmas, and the name is maize bushy stunt. It was discovered in Mexico, but there is no information if that disease, or there was no information if that disease was present in native varieties of corn. And this is important because probably many of you know that Mexico is the center of domestication of corn. So they have a lot of native varieties. So I went to small communities, and luckily for me, but bad for the farmers, we found a lot of symptoms related to phytoplasmas. They were positive. We found we identified the strain, the species, we identified the group, and we also performed a study about the social impact that this disease could have for small communities. Then later, I tried to identify the insect vector of the pathogen, and something really interesting is that we found a new leaf hopper that was not only positive for phytoplasma, but we identified that it was also reproducing in native corn. So this was a very productive project that was a part of my PhD. Then during my second visit to Saskatoon, the first one was to do part of my PhD project, molecular analysis, PCR, working a little bit also in the description of a new marker to characterize phytoplasma. But the second visit was more focused on this disease. The name is strawberry green petal disease. You can see the symptoms and why the disease is called like that. This was in collaboration also with an American company that has offices and a lot of people working in Mexico. The name is Driscoll's. We wanted to develop a diagnostic method to detect this phytoplasma. Why? Because they noticed that the transmission was happening through nurseries. So they wanted to establish a quick diagnostic that will be able to stop the spreading of the disease. So for that, we developed ECID, which is a lab assay. We lyophilize all the mix of the components, the polymerase, the primers, everything we lyophilize in the tube, and then we just add water and add the DNA, and we will be able to know really quick if the pathogen was or not in the plant, even before the symptoms were shown. So this was with Agriculture and Agri-Food Canada, and this diagnostic is right now in the process of being, this is published, so it's not nothing secret, but it's also in the process to being uh, make it commercial with Harvest Genomic. Is a company in Ontario. Later, something that was also added to this diagnostic is that we developed a really quick DNA extraction method using FTA cards. So using these cards, we are able to just smash the insect, also smash a leaf, and then take a section of that circle and then extract the DNA. We have been able to do this in less than 30 minutes, know if an insect, a plant is infected. So this time in AFC was also really, really productive. So I was really happy with that, but I was not able to stay as a postdoc. So I had to go to US for a year, but I really wanted to come back to Canada. I fell in love with the way that we work here with the scientific community. It's quite different to the States. If you have been there, you know. So I wanted to come back. That's why when I saw the opportunity to come back and work with clubroot, I applied immediately. I knew about clubroot a little bit because being in Saskatoon, a place that grow a lot of canola, you hear about the disease, but I really had no idea what it was. That was my expectation. Is a club in the roots? No, it's not like that. If you, many of you, English is your second language, so you probably are laughing at it. But club is also galls. So the disease is characterized by the presence of galls or clubs in the roots. This disease, clubroot or Lernie de crucifère, is a devastating disease affecting cruciferous crops worldwide. This is not a problem only in Canada. This is a problem in Australia, in Asia, many countries in Asia, many countries in Europe. So it's a big, big problem, and there is a lot of people affected by this disease. Of course, it's here in Canada. The prairies, Manitoba, Alberta, and Saskatchewan are the main affected because canola is a big crop for this province. And it is caused by the soil-borne, unculturable, intracellular biotrophic protist, Plasmodiphora brassica. So this pathogen has a lot of characteristics, as you can see. But interesting that this pathogen was discovered in the 1700s, but still today is a big mystery. So something really interesting about the disease is that the presence of Plasmodiphora brassica in a susceptible host turn the roots into a nutrient sink. So you can see here the original symptoms. For example, these are canola roots. You can see how it looks affected. I should have put a picture of the non-infected, but just imagine that is a line, thin line with a lot of root hair. So it affects a lot of crops. You can see here some example, cabbage, affect canola. But what the pathogen needs to do to turn the roots into this nutrient sink? Well, the pathogen needs to manipulate the cell cycle of the infected tissue, and there is evidence that the infected cell start to go into endoreduplication, meaning that the cell cycle is not working properly and the cell start to grow. There is evidence in the plant side also affect the transport of the infected cell. We have that in the infected cell, the cell start to recruit more transporters than in the non-infected cell. This is really interesting, and again, the evidence that we have is in the plant side. Then also infect the plant metabolism. We know that infected cells also change the starch production. For example, this is a compound that is normally produced in the plant as a defense mechanism, but also change the amount of sugar that, or different distribution of sugar that the infected cell produce. And of course, the pathogen also needs to manipulate the plant immunity, because the plant should be able to fight a different pathogen. But when we see the symptoms and we see the full cell life cycle of the pathogen complete, then it's because the plant cannot fight the pathogen. For all this, we basically have evidence only from the plant side. In the literature, there is only evidence in the plant side. We still don't know how the pathogen can do all these, I call it manipulations. So one tool that the pathogens use to do this are effector proteins. So effectors is a term that can group a lot of different molecules that the pathogens use to infect the plant, to manipulate the plant host. But there is a lot of focus in the proteins inside this group of molecules, because there is a lot of evidence showing that are really important for all these processes. There is a lot of information about omicet and fungus effectors, bacteria effectors, and also lately there is a lot of evidence about nematodes also using effector proteins to manipulate the host. But what about protists? This is a really small group, not many, we don't have many protists that are also plant pathogen, and this might be a reason why the research haven't been focused on this. Well, to this day, 2020, we only have one full characterized effector, or very well characterized effector for Plasmodiaphora brassica and for protists. And it's a methyltransferase, which can methylate salicylic acid. Is a molecule really important for signaling in plant and defense signaling in plant. So there is a lot to know about protists and about Plasmodiaphora and the effectors used by this kind of pathogen to infect the plant. When I started my postdoc at the University of Saskatchewan, they performed the RNA-seq for secondary infection. And now it's something that I need to clarify. This pathogen, the infection is divided in primary and secondary. And why secondary infection is so important is because it's the moment when the pathogen induces the galls. So there is evidence that some resistant plants can allow the penetration of the pathogen to the cell, but then doesn't happen the secondary infection. So secondary infection is really important to really fight the pathogen. So what we wanted is to identify effector proteins. So after performing a bioinformatic analysis, putting a lot of restriction about size of the protein, be sure that the proteins had signal peptide, meaning that probably is going to be secreted, no transmembrane domain. And also we try to identify some domains, characteristics of effectors in other evolutionary groups. So at the end, we ended with 32 candidates. Not a lot when you compare to omicets or fungi, but just when our study was submitted for publication, another paper came out from a different group trying to identify effectors in the primary infection, and they also identified 32. So we were kind of happy we were not far from what could be the truth. But among these 32, we had at least 6 or 7 that they had a domain that we had an idea based on in silico analysis of what they could be. We have kinase, serine protease, glucanase, translocase. All these have been are domains that have been identified in effectors in other evolutionary groups. So we wanted to focus on this. Mainly, one of those was really interesting for me, and it was a cysteine protease inhibitor. And why was really interesting? Well, the apoplast is the space between the cell wall and the membrane, also known as the first battlefield for plant pathogens. Many interactions happen in this part, in the apoplast. One of those interactions that have been identified in oomycetes, nematode, fungus, bacteria, and virus is the interaction between a pathogen cysteine protease inhibitor and a plant papain-like cysteine proteases. These have been conserved in all these group of pathogens, but there is no evidence about the protist. So I was really interested to try to identify if that effector candidate that we detect was indeed part of this mechanism. So something also really interesting is that when I amplified the gene that codified for this effector, it was 100% identical in different pathotypes, but also in Canadian pathotypes, but also was identical to Europe and China, a pathotype from those countries. So this was really encouraging. Whatever is happening, it was conserved among pathotypes. Then in silico analysis also showed that could have a functional synaptide, that could be an effector, and that could be apoplastic. So when we did the 3D modeling of the protein, we also identified that the structure is very similar to other cysteine protease inhibitor from other pathogens. And the alignment also showed that the N-terminal domain was conserved and the loop one was also conserved. And it's really important, these are really important for interaction. We didn't find that the loop 2 was completely conserved, but still we wanted to see if it was really a cysteine protease inhibitor. So during some analysis, I was able to also identify that the synaptide was functional. The first that I had to do was to obtain the protein. It's a really small protein. And then I perform an activity-based protein profiling. So for that, I use papain as a model cysteine protease. Then I use the chemical inhibitor E64. Then I use the recombinant protein, and I use DCG04, which is the inhibitor, but biotinylated, in a way that I would be able to, using streptavidin, to recognize biotin if papain was attached to DCG04, but not it was inhibited by the chemical or by the recombinant protein. So this is simple. I'm going to explain right now. We have 4 tubes. We have papain in all the tubes. Then I pre-incubated one of the tubes with E64. The other tube was with the recombinant effector or candidate effector. And then these 3 tubes, I incubate them with DCG04. So the signal was only detectable in this tube, which is kind of the positive control. We have papain and only DCG04, but I couldn't detect any signal in these tubes, meaning that papain was already inhibited by here the chemical and here by the recombinant protein. So this was a really simple experiment that allowed to know it was really a cysteine protease inhibitor. But of course, I wanted to answer other questions, like for example, if it's apoplastic. Well, what I did was to fuse the protein to GFP and then infiltrate agrobacterium, using agrobacterium, in a canola cotyledon. And I found that indeed, after separate the membrane from the cell wall, the signal remained in the apoplast. A similar result we obtained with Nicotiana bentamiana. So you can see here, this is the membrane, this is the cell wall, and the signal remain in the apoplastic space. Then, to be able to confirm if indeed was inhibiting cysteine proteases from the apoplastic from Arabidopsis, I forgot to mention that Arabidopsis is a member of the Brassica family, so is a host for Plasmodiophora brassica, luckily for us because Arabidopsis is the model plant for molecular biology. So what I did was to the same experiment, but now using apoplastic fluid from Arabidopsis roots. So I obtained similar results indicating that SPVP53 is a cysteine protease inhibitor that inhibits Brassica's cysteine proteases. So something I was also interested is to know the target of this cysteine protease. So I first studied the expression of cysteine proteases in infected and non-infected plants. And in all the cases, we observed a downregulation of cysteine proteases. Then we study only to see how much active cysteine proteases we could detect in infected plants. And in every case, we did this for 5 different hosts, the amount of active cysteine protease was lower compared to the non-infected plants. So we have here a mechanism, something is happening that is downregulating the expression, but it's also reducing the amount of active protein. So the next step was just to see with whom is interacting. So I select these 4 because they were like the clearly more downregulation of the expression. And I obtained them with the GST tag. I mobilized those proteins in a column, and then I pass the effector through the column to see with which one of those it would be co-precipitating. And we found that it was co-precipitating with 2 different proteins. Here is really faint, but we found some is XCBP3 and XCP1. So these 2 proteins are really interesting because they are xylem-related proteins. And there is evidence that the pathogen induced xylem disruption during the infection. So there is a lot to speculate here, but we needed to really do more experiments. As I mentioned, we cannot knock out any gene in Plasmodiophora, but we can do it in the plants. So what I did was to explore how different mutants for cysteine proteases behave during the disease progress. And it was really, really interesting to see that XCP1 mutants plants remain in the medium-sized goal category during the infection at the end of the secondary infection, while other proteins, other cysteine proteases like RD21 or RD19, they just behave as the core 0, that is the wild type Arabidopsis that we use in the community. So this was really interesting. Is this interaction really necessary for pathogenicity, or is delaying the infection because the xylem is not completely developed? Those are still questions that we need to answer, but something that we also were able to see is that the spore formation of the pathogen was also reduced in the XCP1 mutant. So I expand a little bit in this subject because a big part of these experiments were already performed here in Edel lab at Université Laval dans la belle ville de Quebec. I'm in love with Quebec, I have to say that right now. So, but many of the experiments I mentioned before were performed here because we submit the manuscript for a publication, but the editor and the reviewer requested more experiments, and it was just at the beginning of the year with the transition to the lab. But this is something that we probably the group in Saskatchewan is going to keep investigating what is exactly the role of this interaction between the effector, the cysteine protease inhibitor, and the cysteine protease. But my lab is going to take a little bit different direction. And this is a scheme that represents my lab right now at the moment. This will change for sure because we are interested in pursuing other pathogens, other research. But at the moment, based on the grants that I was able to apply this year, we are going to work with phytoplasmas, mainly in blueberries and in strawberry here in Saskatchewan, sorry, here in Quebec. There is a problem with a blueberry stunt phytoplasma and with a strawberry green petal phytoplasma. What we plan is to focus on the vector because our thesis is that the vector can be used to trace climate change. So this is something really interesting. We presented 2 projects. Let's see how it goes. The other subject is going to be Ralstonia solanacearum. So this is a bacteria that affect solanaceas, and one of the main problems is with potato. Luckily for us, we don't have Ralstonia in Canada or in Quebec, but this is a collaboration that will happen with Cuba. In Cuba, we have a lot of different races of Ralstonia solanacearum, and Cuba is a client of Quebecois potato companies. So I found a connection there, and we can benefit knowing how potato is from Quebec is going to react to different races of Ralstonia solanacearum. So I'm really excited for this. Hopefully we get that project. But the main focus is going to be Plasmodium, Plasmodiphora brassica, and canola. So Plasmodiphora brassica is really hard to manage. Why? Because the resting spores can survive up to 20 years in the field. And I mentioned that the main problem is in the prairies where it's really, really cold. So that tells you that Plasmodiphora can survive also really harsh winters. So it's no way that it's going to go only because it's cold. To this day, we don't really understand the resistance to clubroot that is available in the market. For example, we have several canola hybrids that several companies sell, but they don't know how they work. What is they have an idea of the resistant gene in some cases, but many of the cases they don't know. And the other big problem is that, for example, I buy these resistant germ seeds, I planted in my field, and after 1 or 2 years, the plants get infected. So the strategy is really, really difficult and costs a lot of money. So one of the big things that we really are missing in the research is a new source of resistance. So we need to have new sources of resistance that we really understand, and we can then have a stable material in the field. But how are we going to do that? And this is basically, in a nutshell, my research program. We want to identify new sources of resistance. But what I'm proposing is going to the basis of plant immunity. So plant immunity can be represented by this graph. We have a first layer of plant immunity that is known as a PAM trigger immunity, and PAMs here in the bottom is a pathogen associated molecular patterns. So these PAMs are those molecules or structures that are unique to the pathogen. An example is the flagella in the bacteria. It's recognized by plants, and it elicits the PAM trigger immunity. So you can see in this graph that this immunity is the first layer of immunity, which can protect to certain group of pathogens, but it's not as effective. You can see here the amplitude of the defense. And then we have a second layer of immunity that is triggered by effectors. So I mentioned already that effector is a really broad group of molecules, but we are going to focus in proteins. So effectors, when they recognize the R gene resistant protein, resistant gene, resistant protein, they are called avirulence. So they are called avirulence proteins. But for this presentation, I'm just going to refer as effectors because it can get a little bit complicated. Something interesting about the ETI or effector trigger immunity, you can see here how the amplitude of the defense is really high. It goes over the hypersensitive response threshold. And other really interesting aspect of ETI is that, for example, if the effector have more than, if the pathogen have more than one effector that is recognized by a resistant protein, then is longer the resistance and is more stable and is stronger. So this is something really interesting. And indeed, ETI is the base of a resistance in a plant, in a long-term resistance. So something I also want to tell you, and it's going to be also the base for my research program, is how the pathogen resistant protein and the effector interact. So we have here that, for example, once the effector can reach the target, the plant is susceptible. So that's something that we have to know. But when the effector then interact with the resistant gene, that I have to clarify something. In the last year, NLR have been identified as the primary family of resistant proteins. So you are going to hear in the presentation a lot of NLR. So this means resistant proteins. Anyways, so once the effector interact with the resistant protein, we have resistance, of course. But this interaction is not always direct. We can have guard, we can have decoys, and we can have a lot of different mechanisms. So, of course, we have a arm race. We have a pathogen evolving a lot of strategies to avoid this interaction with the R protein, and we have the plant developing a lot of strategies to try to reach those effectors and then be resistant. So it's a lot of things that we need to know, but something that we can learn from this is that what about we can use the effectors to catch new resistant genes. So, but for that, we need to deliver effector by effector. So here, this is my thesis. Use effectors to try to catch resistant proteins. So the first thing we need is to identify which one are the best candidate effectors to use as a bite for the resistant genes, resistant proteins. So we want to first identify the core effectors for the pathogen. How we would do that? Well, I plan to use a comparative genomics for that. We have a problem. To this moment, we only have less than 5 or around 5 really good quality genomes. None of them is Canadian. So to fix that problem, I want to sequence a single spore isolates collection. From here, from Canada, we have from Manitoba, from Alberta, Saskatchewan, and Quebec. But I'm also thinking in expand this to other countries to really have a big representation of genome and being able to say, this is the core effectors, and these are the effectors that might trigger effector trigger immunity. So we have also Norway, Denmark, Sweden, Denmark again, sorry. It was don't remember, but it was another, the Nordic country, because I have a collaboration, but also Mexico. To this day, there is no genome for Latin American isolates. And we recently published a paper saying, well, identifying that in Latin America, there is a big problem with clubroot. So it will be really interesting in terms of diversity to have the genome for all these isolates. So once we have the core genomes, then my second plan is to try to identify effectors eliciting effector trigger immunity. But how to do that with a pathogen that we cannot culture? So what I'm proposing is to use a surrogate. So I want to use a bacteria pathogen to Arabidopsis, then transform this bacteria is a Pseudomonas syringae with Plasmodiophora effectors. Then it will deliver one by one the effectors. At the beginning, the screening is going to be with around 60 to 100 effectors. I'm going to focus in those families that previously has been identified as elicitors of ETI. Of course, these have limitations. And one of the main limitations is that I'm using a prokaryote to express eukaryote proteins. So we will have to be careful with which proteins are we selecting, because if we have proteins with a lot of cysteine bridge and a lot of different modifications, will be not the best option. The second limitation is that I'm using a leaf surrogate, a leaf pathogen, as surrogate for a root pathogen. This is also a problem because maybe the R proteins are not expressed in the root, and we are finding results that are not going to translate to Plasmodiophora brassica, but we will think about it, and we will have to confirm that the results can be translated to the roots. Then we will be detecting ETI. What we are going to do is to measure at least the results in a big collection of Arabidopsis thaliana ecotypes. So this is something that we already started. We have a lot of ecotypes available in a database, a collection, sorry. Then what we are going to measure is hypersensitive response, but we are also to check how the presence of these proteins are affecting the bacteria growing. So this is really interesting because maybe we don't see hypersensitive response, but we see that the presence of this effector is promoting the growing. So it could be a positive effect for the pathogenicity, but not in the way that we are expecting. So this is something that we want to also pursue. But then the next step will be to really identify which one are those NLR and LR mediating ETI. What I'm proposing for that is to use Turbo ID. So what I'm going to do is to then the factors that we see that they are eliciting ETI, We are going to clone them and transform the plant fused with the turbo ID. What is turbo ID? Turbo ID is a promiscuous biotin ligase, meaning that using biotin, I'm going to be able to tag all the proteins around the effector. This is, for me, was really, really important because, as I mentioned in the previous slide, the interaction not always is direct. So we will have that some are interacting with the R protein, but some are interacting with the guard, with the decoy. So using this ligase, I will be able to identify not only the R protein, but also the mechanism that the effector is using to interact with the R protein. So once I have the protein purified, we can go to mass spectrometry and try to identify which one are those proteins from the plant that interact with the effector. So the next step of my research plan, and this is only for the hopefully 5 years, is going to be the integration of those results that we obtain with Arabidopsis in other crops, economically important crops, mainly canola. So we want to see if, for example, those new R proteins genes are present in other brassicas. We want to see if we can transform a canola, for example, a resistant canola, if we add this, how they can behave in time. Then a susceptible canola, if we put this gene, how behave in front of the pathogen. Is kind of ambitious, but I think it can go further than 5 years. So what I have been doing in this time? Well, requesting plasmids, bacteria strains, seeds. This takes a lot of time in normal time. Now in COVID time, it takes longer. Acquire antibody and supplies, writing grants, recruiting students, which is also really slow now with COVID. And I have to say, I'm looking for students. So if you know somebody. But I also was interested in answering this question: how Plasmodiophora escapes PTI. Well, it's clear that the pathogen can escape PTI because it goes and develops the full life cycle, and we see the goals and everything. So, but how is happening? Well, something really interesting is that Plasmodiophora spores, the main component of the cell wall is chitin. And chitin is a very, very well-studied pump. So it's a pathogen-associated molecular pattern. But even when it is well recognized by the plant, it can go through and is undetectable. How the pathogen does that? Well, looking in the genome of the pathogen, I found that it has a lot of proteins that are related with chitin. So we have some proteins that they only have chitin binding domains. Others are deacetylase, others are chitinases, and I even found a tyrosinase that has a chitin binding domain. So this was really interesting. It has a lot of protein, so it probably is doing something to avoid to be detected. So the first thing I did was to check RNA-seq data that was previously developed by another group, and it was for 3 different hosts and in 3 different development stage. We have here a germinating spore, a mature spore, and plasmodium. As you can see, the expression is really different depending the host, which is really intriguing. So the pathogen adapts depending the host and the host amount of different chitinases or different proteins and mechanisms, but it's also different in this live stage. Of all those proteins, I was really, really interested in these three, that they only have one domain, one chitin binding domain. So we had that three of them had only one, and the fourth one had two. But I focused in those three as a model to see if really they could bind chitin. The first thing was also to confirm if they are secreted, if the signal peptide is functional. So with this assay using a modified yeast, I was able to detect, putting the signal peptide here, it will secrete this invertase, and then this transparent compound will be reduced to a red color compound if the signal peptide is functional, because it will secrete this invertase. What I found is that indeed, this protein had a functional signal peptide. The next thing I did was to study the domain. So it was really interesting for me to find that it is a carbohydrate a binding module 18, which is not common at all in plant pathogens. It has been only described before in amphibious fungi pathogen. So this was really interesting, but it doesn't surprise me either, because a previous study found that Plasmodiophora is just in the limit of the separation of fungi and protists. So it's really kind of old pathogen. So what I did was to check all the conserved amino acids, and I found something interesting. Here, for chitin binding protein 2 and 1, there is a new amino acid here, and here the glutamine conserved is not the glycine, it goes to glutamic acid. So taking that into account, I said, okay, let me just go with two of them instead three. And what I did was to express two of them, the protein, recombinant protein, and then test if they bind chitin. What I found was really interesting. The Plasmodiophora chitin binding 2 was able to bind chitin bits and chitin, and the 4 was also able to bind chitin bits and chitin. They were not able to bind cellulose, chitinol, chitosan, or silane. So this was really interesting. We know that they really bind chitin, but I wanted to know a little bit more. I wanted to know why the pathogen is using, or how the pathogen is using these proteins. So what I did was to study the expression using time points from 0 to 28 days post-infection. And I found that one of them was overexpressed 2 days post-inoculation during the primary infection, and the other one was overexpressed during the secondary infection. The second was only overexpressed during secondary infection. So where is that in the life cycle of Plasmodiophora? Well, we have here that 2 days is around when the resting spore is passing to zoospore. But then the secondary infection, that is this around 21 days, is when the resting spore is being formed, is the formation. This was really, really interesting for me, because you can see that it's kind of, I call it guarding the resting spore, guarding the next step of the resting spore, but also guarding the formation of the resting spore. This was really interesting. Then what I did was to see if they can really directly bind resting spores. So I use a really high concentration of resting spores, and I incubate them first. For example, here, spores and the protein, we can see that they precipitate in the pellet. Then what I did was a spore incubated with the protein, but then I add a chitinase, and then still I see it here in the pellet. But when I do the different order, I go from a spore, chitinase, and then the protein, I was not able to detect it in the pellet. The same happened with the other protein. So this is evidence that the chitin binding proteins might be really important to guard the spores during the infection and during the formation of the spore and coming to the soil again. Still, I wanted to also see if they are able to mask the pathogen and to avoid the response from the plant. So we have here that when the receptors, they detect chitin here in red, they phosphorylate MAP kinases. And then that's a really long mechanism, but at the end, we have the defense. So this is PTI, how it happened. Well, really, in a nutshell. So what I did was the following. I used the plant. I put the plant here only in water. I put the plant here in chitin. Then I put the plant here only in our protein. And then I put the plant in chitin that was pre-incubated with the protein and try to detect the phosphorylation of the MAP kinases. As you can see here, we see that the response was reduced when I pre-incubate the chitin with the protein, meaning that yes, it can be masking the effect. Similar, but it's not that clear. We have the response here is slower, lower, but still not perfect. But if you remember 2 slides before, this protein was not able to bind as strong as the number 2. But still, it's a lot of speculation. We will have to do other analysis like protein characteristic of the protein, how is the interaction and all that. But what I'm hypothesizing is that chitin, these proteins that bind chitin here in green, they not only protect the spore, but they also might be attaching any chitin that is being released during all these transitions that happen in the apoplast and in the cell. So this is basically what I've been doing for all this time. And in my lab, there is opportunities. Hopefully, by the end of this year, I will have grants coming, and we need students. So if you know anybody that wants to do a master or a PhD, please send it to me. And of course, I want to thanks for the invitation. Sorry for, I think it took too much time. And I want to thank Richard Belanger Group, everybody, Caroline, Chloe, Vanessa, Amandine, Richard, of course. I want to thank Francois, Valerie, Charles, Dominic, and everybody in the department, in CRIF, in the faculty. Everybody has been really, really awesome. Thank you.
All right. Thank you, Edel. Do you have any questions for Edel?
Maybe I have a question about your future research directions. So I understand from your talk that it's very difficult to do kind of classical genetics with your pathogen because it's very tough to manipulate. But I know that there are more and more tools to do genetic manipulations and Arabidopsis. So are you planning to also study kind of these issues and how these proteins affect the host organism from that angle, or you're more focusing on the proteomics for the moment?
That's a really good question. And yes, one of the things I'm planning to do is try to transform Arabidopsis with these effectors. It's going to be a parallel line to see how, for example, how when I transform with the effector, for example, do RNA-seq and see what is happening, what is changing in the host. Some of these effectors might be really hard. We will have to do with the what is the name? This system that we have to put a chemical inducible system, because we are hypothesizing that many of these effectors are going to have, for example, one that we found is a cyclin, and we think that might have an effect in the cell cycle of the plant. So when I tried to have a transgenic plants, I had 0. I think that is a deleterious effect. So we will have to have a lot of inducible systems and see at that moment what is happening. But yeah, of course, I want to do it, but in parallel. Like, for example, can be a lot of project for students to get like 5 effectors, put it in the plant, do RNA-seq, see what is happening. Yeah, it's really interesting. There is a lot to do. In the genome side of the pathogen also, I want to answer many questions, because we have no idea how is the real structure of the genome. We have no idea of the transposable elements, for example. I'm really curious. I did some search. We found kind of new transposable elements, probably. So I mean, everything we do with this pathogen will be really interesting.
Thanks a lot.
Thank you, Philip. Other questions for Edel?
Yes, I have a question.
Please go ahead.
First thing, thank you for your presentation. And I read your article about the transcriptome, and I saw your cutoff the proteins that they have about 400 amino acid genes that encode proteins with 400 amino acids. And I also read Lopez et al. That is quoted in your article. She works with 300 amino acid proteins, genes that encode the proteins with 300 amino acids. And why do you choose to work with 100 more amino acids?
Oh, it was just really trying to get more. We were like with so little amount of interesting candidates, and we decided, let's go with more, because this pathogen we don't know. And I thought that would be nice to see. there was no like too much thinking, more than try to get more interesting candidates. But in the literature, yes, it's from 300 to 350, what they consider small secreted proteins. So I went a little bit further, and in this last analysis, I went even bigger. I was like 500, and I was able then to get all these chitin related proteins, because many are small, but I thought maybe we are missing some interesting, and still they have a synapeptide that is functional, and it was something interesting to check. But there is no, I don't believe that there is really something set on a stone about what do you can choose.
Okay, thank you, because I'm kind of doing the same thing with other organisms, and I read our work. It adds a lot in my work, and thank you very much.
No problem. I think that depends what do you look for. I would say.
I'm looking the same thing of you. I'm work with host and pathogen interaction, and I'm looking the same thing, and maybe we can talk after the meeting too.
Yeah, perfect.
Thank you.
Reach me and my email. My webpage is not working. Sorry for that. I'm really bad with technology.
Okay, thank you.
Other questions?
Yes, I have a question.
Oh,
Please go ahead.
So one of the factors that is usually used also to screen for a factor is cysteine content. And it looks like you didn't use it in your screen. So is there a special reason?
yes, we did. We did. I tried to define what was a cysteine rich. So some people say 5% of the content. It has also it's kind of the range. So I looked for it and I was able only to find like 5 or 6 only that we considered that were like cysteine rich. I think I used I think it was like 6 point something percent of the content, 6% of the content of the number of amino acid that was cysteine. Yes, I think we have it in that manuscript. We have it in the supplementary information. Where not many, and I right now expanding the size of the search. We have, I think, less than 10 that we consider that they are small and cysteine-rich proteins. You know, this pathogen, because it's so different, have been really difficult. I recently had also a meeting with another scientist from Austria, and she was telling me the same. She told me that they work with omicets, and the number is always so high of candidates, and you know what to look for, and you have the domains to look for. But with this pathogen, it's really different.
Maybe another question also. Does the origin, the geographic origin of that pathogen known or it's unknown?
That's a really good question, and I also would like to work on that. I think it's now with this opportunity to sequence a lot of genomes, we would probably have the opportunity to answer those questions too. The first report says Russia. So that's the first report, but to this day, there is no genome or any information about Russian isolates. So what we have is also Germany. We have also that could be in all these Nordic countries. So we believe that it's in that area. Here to Canada was detected for first time in the 90s in Quebec, indeed, was in Quebec for first time, and then in 2005 in Alberta.
And what about the genetic diversity of this organism?
That's a really good question too. So the taxonomy of the organism is really messy because we have what we call pathotypes, but that has been only really explored and expanded here in Canada. In Europe, for example, they just call it 1, 2, 3, something like that. Here lately, we have 30 different pathotypes that we call 6A, 6B, and it's only based on the response to the host. So we don't have really any genetic study that is really strong. And that's why I'm again proposing to sequence a lot of genomes, because it's the way that we will really know how is the diversity. There was recently a study published by a group in Ontario about that, the population studies and all that based on genome sequencing, but the sequence were not the greatest. They had like basically only 5x coverage, and it's hard to really draw conclusions.
Thank you.
No, welcome.
All right. Maybe one last quick question, if someone has a burning one. No. Otherwise, merci beaucoup, Edel. C'est une super bonne présentation. Even for non-specialists, we could see the importance of the work and also the nice biology that you're going to be doing. That's great. Thanks a lot.
No, and well, if you know students, send it to me.
Yeah, definitely. All right. Bye-bye. Thanks,
Bye.
everyone,
Thank
for
you.
coming. Bye-bye.
Thanks for the invite.